Bismillah ar-Rahman ar-Rahim. The title of our lecture is Transplantation, and this includes transplant immunology, terms used in transplantation, tests used before transplantation, immunologic mechanisms, types of rejection, and treatment. First, the transplantation is an imagination. They did not know that it's become true, and they imagine that the transplant the leg. This is the black one, and this is the white one, and this is the transplanted lower limb. And there is a terms in the transplantation. First, we have the autologous, which is transplantation of autograft from one side of the body to another side in the same body. There is no rejection. Best example is the skin graft. And the another term, which is syngenic, which is a transplantation of isograft between genetically identical twin. This is identical twin, and there is no rejection. And the another term is allogenic, which is transplantation of allograft, which is homologous graft between genetically different individuals of the same species, and this is result in rejection. This is the same species, human to human, or genetically, which is different. And xenograft, or it's called heterologous graft, which is a graft transplanted between individuals of different species, between animal and between the human. And this is a result in rejection. So rejection occur here, and there is no rejection in the first and in the second terms. The fate of any allograft transplant, either accepted or rejected. The causes of rejection: the recipient immune system looks to the graft as a foreign substance, as a foreign infectious organism. And this is achieved by looking to certain molecules on the surface of the leukocyte, which is called the human leukocyte antigen or histocompatibility antigen, and it's called so because it is responsible for the rejection in transplantation. So it is called histocompatibility antigens. The HLA antigens, each graft transplanted from one member of species to a different non-identical member of the same species. Example, one human to another are termed allograft. Graft, the organ that's transplanted, whether tissue, whether cell, whether organ. It's called allograft. In this situation, the two individuals differ in the HLA typing in the major histocompatibility complex, which is the same name, whether MHC, whether HLA, and also known as human leukocyte antigen, and or differ at minor histocompatibility antigen. As you know, we have major histocompatibility and minor histocompatibility antigen. HLA antigen can be divided, as mentioned in the previous lecture, class 1, class 2, and class 3. And this is the presence, class 1 and class 2 presence on the antigen presenting cells, and can be upregulated on vascular endothelial cells after ischemia, perfusion injury. The immunology of allogenic transplant: first, it is cell-mediated immunity, which is play a key role in allograft rejection. The T cell, which is responsible for the cell-mediated immunity, T helper cell, is designated by CD4, is stimulated by allo antigen plus the MHC molecule. As you know, each antigen, which is presented on the MHC, which is expressed on the graft, and is divided into two phases: sensitization phase and the effector phase. The sensitization phase, which is antigen reactive lymphocyte T cells plus T cell plus T CD8 positive of the recipient proliferate in response to alloantigen of the graft. So there is a proliferation of the T cells. And the effector phase, which is the immune destruction of the graft, which is occurred by the cell-mediated immunity. Innate immune response, which is inflammation from ischemia, reperfusion injury in the allograft leads to migration of NK cells, macrophage, neutrophil to the allograft, resulting in injury. The adaptive immune response, there is a stimulation of CD4 positive cells, which is primed against donor antigen and release the cytokines. As you know, after stimulation of T cells, there is a release of cytokine, which direct an immune response in the allograft. This results in stimulation CD8 T cells, T cytotoxic, macrophage and NK cells, and migration to the graft, causing cell damage and apoptosis of the graft. The cytotoxic CD8 T cells kill their target in an antigen-specific method through two methods: direct cell and cell contact in one of two mechanisms. And this is first by secreting proteins, granzymes, and perforin, which is a result in cell lysis. And the second mechanism by the activation of the Bas-Bas ligand pathway. As you know, this is the mechanisms of the T cytotoxic. The Bas ligand in the T cell binds to the Bas on the graft cell, inducing apoptosis. At the same time, CD4 T helper cells activate these cells via the CD4 T ligand, and this is all the mechanisms of T cell and B cell activation, as mentioned in the previous lectures in the previous years, which help B cells to transform into antibody-producing plasma cell. As you know, differentiation of B cell to plasma cell that lead to secretion of the antibody, and antigen antibody lead to complement activation and lead to destruction of the graft. As you know, each antigen antibody immune complex lead to complement activation. And the T cell recognize the MHC peptide complex through the heterodimeric T cell receptors expressed on the cell surface, and in the normal host, T cells are trained to recognize foreign peptides expressed in the context of the cell MHC. This is a review of the T cell activation and how we recognize the foreign antigen, which is the graft antigen. Same mechanism, it's not different. And molecules and are tolerant to self-antigen. As you know, the T lymphocyte tolerant to self-antigen through the thymus education. And T cells also recognize for MHC peptide complex, and this is now known to occur through two distinct but not necessarily exclusive pathways, the direct and the indirect allo recognition pathways. So the T lymphocyte recognize the allograft antigen by two mechanisms: the direct and indirect allo recognition of the foreign allograft antigen, which is the MHC plus peptide molecules, which is expressed on the donor cells like kidney cells, lung, heart, or whatever, which is in a process unique to transplantation. A T cell receptor CD3 complex along with T cell surface molecule CD4 or CD8 interact with its counterpart MHC molecules and antigenic peptide. This is all, as you know, before the T cell activation. And there is co-stimulation. Positive co-stimulation is required for T cell activation. As you know, the previous is the first signal of T cell activation, and this is the second signal of the T cell activation. There are numerous positive and negative co-stimulatory pathways, and best non-positive co-stimulatory pathway, which is the interaction of CD28 on the T cell surface with the B7 molecule, which is found on the antigen presenting cell. As you know, this is the second signal in the T cell activation, which is you mentioned in the previous lectures. The combination of T cell receptors, ligation, and co-stimulation result in T cell activation and production of various cytokines and growth factors like interleukin 2. Interleukin 2 bind with the CD25 activation of T cell receptor and positive co-stimulation result in the production of potent T cell growth factor interleukin 2 and upregulation of activated interleukin 2 receptor CD25. Interleukin 2 works in an autocrine and paracrine manner on the CD25 receptor to induce lymphocyte proliferation. This is the mechanism of the interleukin 2. And the end result of T cell activation is rejection and the effector phase of the immune response. All previous explanation is sensitization phase, and this is the second phase, which is the effector phase, which is the rejection. And the effector phase of the immune response exhibits memory and escalating response to foreign antigen. It requires secondary lymphoid organs where antigen presentation takes place, as well as both the innate and the adaptive immune system. So to avoid rejection, patient must be sent to the following immunologic laboratory test, which is first AD of the group, second tissue typing A, B, C, G, and the RDQ. Cross match, which is very, very, very important, between donor lymphocytes and recipient sera. Two, this test is to detect the presence of antibodies against the lymphocyte of the donor. Another test, frozen lymphocyte trait of T or PRA, panel-reactive antibodies, procedures for T or T and B cells for detection antibodies in patient serum. This is monthly done to the patients. And anti-HLA antibody screening using either Luminex cross-matching assay. Previously used serological test and nowadays use machine which is called Luminex Luminex cross-matching assay. The principle of this Luminex is as flow cytometry. And the C4D staining, the position of C4D in graft capillaries, and this is done by renal biopsy and detection of this C4D. Other cetal and complement. Other tests like virological tests, CMV, Epstein cytomegalovirus, Epstein-Barr virus, hepatitis B virus, hepatitis C virus, HIV, AIDS. As you know, cytomegalovirus, the target organ is kidney. So if the patient is positive for this, so the end result of transplantation is rejection. So the first and most important test is the HLA, which is the battle between the immune cells and the donor cells. Type of donor. In Iraq, relatives, whether father, mother, sister, brother. While abroad, this considered as inhumanity. The type of the donor is cadaver from accidents, car accidents, and brain death. And there is an association between countries, which is European Association for Transplantation. And this includes all countries in the Europe. And the another association, which is a Scandinavian Association for Transplantation. And this society includes all Scandinavian countries like Denmark, like Netherlands, like Sweden, and the another association, which is American Association for Transplantation, which includes North America.